Well, when we talk about glucose oxidation, the first thing that generally comes to our mind is glycolysis and citric acid cycle. But did you know that glucose can also be oxidized through another pathway? Well, let's find out more with this session, which is on hexose monophosphate pathway. Now, before we begin, I'll just take you through the learning objectives of this session. You will need to know the definition, site, substrate, what are the products and key enzymes which are involved in the pathway. You will also need to know the reactions which are divided into two phases, and then regulation and the significance of HMP pathway. So, first of all, HMP pathway is an alternate pathway for glucose oxidation. It is said that nearly 10% of glucose is oxidized by HMP. Now, HMP also is known by other names. As such, it is known as a shunt, HMP shunt. So, shunt, the literal meaning of the word shunt is nothing but a passage which allows movement. So, whatever intermediates or whatever products are formed at the end of the pathway, some of those products are channelized into glycolysis so that they can regenerate and form glucose 6-phosphate, and which can be either reutilized or it can go into energy production. Other than that, there are other two names, that is pentose phosphate and phosphogluconate. These two terms are basically based on the intermediates formed in the pathway. Now, you have to understand that unlike glycolysis and citric acid cycle, where there is energy production, what happens here in HMP pathway is there is no generation of ATP, there is no generation of energy. So, what is the use of HMP pathway? Now, the basic use of HMP pathway is the generation of two important products in our body, which is nothing but NADPH and pentose sugars. So NADPH is basically utilized for the formation of various important compounds in our body, such as fatty acids, cholesterol, steroid hormones. It may be also used in detoxification of certain drugs in the liver, and so on. Whereas pentose sugars, the main pentose sugar which is formed during the pathway is ribose 5-phosphate. Now, this ribose 5-phosphate may be utilized to form one important product which is utilized in the synthesis of purine and pyrimidine nitrogenous bases, which are nitrogenous bases that form the nucleic acids such as DNA and RNA. So, these are the two most important products from HMP pathway. Now, let's have a look at the major organs or the major site where the pathway operates. Now, if you have a look at the major organs over here, you can see that it's liver, adipose tissue, adrenal glands, RBCs, testis, and the lactating mammary gland. Now, the thing is, these organs mainly require NADPH. They rely on the utilization of NADPH for the synthesis of certain important products. Like, say for instance, the liver utilizes NADPH for the synthesis of fatty acids, for the synthesis of cholesterol, bile acids, and also liver utilizes NADPH for the detoxification of certain drugs. So these are all the processes in the liver which utilizes NADPH. Likewise, we have adipose tissue and the lactating mammary glands. These two require NADPH mainly for fatty acid synthesis. And then we have adrenal gland and the testis, which require NADPH for steroid hormone synthesis. And then I've listed RBCs. These are nothing but erythrocyte, red blood cells. Red blood cells utilize NADPH to maintain the integrity of the membrane which encircles the RBCs or the erythrocytes. So these are the organs or the tissues which require NADPH. However, there are certain tissues or certain cells in our body which require pentose sugars also. These tissues contain rapidly dividing cells, so cells which multiply at a very fast rate. So those tissues or organs or sites include bone marrow, Then you have the intestinal mucosal cells, and even tumor cells require a lot of pentose sugars for the formation of nucleic acid because they are rapidly dividing. So again, in those tissues as also, you can have the pathway HMP operating in those organs. So now we'll come to the subcellular site. So we know what are the important tissues or organs where the site operates. The subcellular site is mainly the cytoplasm because the enzymes involved in the pathway are present in the cytoplasm. The starting material could be glucose or glucose 6-phosphate, and then the end product is mainly the two most important products of the pathway, which is pentose sugar and NADPH. And the key enzyme is glucose 6-phosphate dehydrogenase. So now let's have a look at the reactions of the pathway. So as I've said earlier, the reactions of the pathway are divided into two phases. So first we have an oxidative phase where hydrogen is removed from the different intermediates present in this phase. And then we have a non-oxidative phase. So you should remember the oxidative phase mainly contains irreversible reactions. And the non-oxidative phase contains reversible reactions. And the main purpose of oxidative phase is for the generation of NADPH. On the other hand, the purpose of non-oxidative phase is for the generation of pentose sugars as well as hexose monophosphates, which is the name of the pathway. So these hexose monophosphates are carbohydrates containing 6 carbon atoms with a phosphate group, one single phosphate group. That includes glucose 6-phosphate and fructose 6-phosphate. So this is the main purpose of oxidative as well as non-oxidative phase. Let's have a look at the first phase, which is oxidative phase. So, as I've told you, the reactions may start with the starting material, which could be either glucose or glucose 6-phosphate. So, I'll just start with glucose now. Glucose in the first step undergoes phosphorylation in the presence of the enzyme glucokinase, which is present strictly in the liver, or hexokinase. Kinases are known to transfer a phosphate group to the substrate, which is glucose here, and the donor of phosphate group is ATP. So the phosphate from ATP is transferred to glucose, and phosphate gets attached to the 6th carbon atom of glucose, leading to the formation of glucose 6-phosphate. Now, this is the first step. The next step is glucose 6-phosphate undergoes a process of dehydrogenation where the hydrogen is removed from glucose 6-phosphate. And we have the key enzyme, which is glucose 6-phosphate dehydrogenase, which comes into action over here. So the hydrogen acceptor in this step is NADP. NADP accepts the hydrogen from glucose 6-phosphate and gets reduced to NADPH, which is one of the most important products of the HMP pathway. Also, during this step, a water molecule is added, leading to the formation of the next intermediate, which is 6-phospho-gluconolactone. So this is the next intermediate which is formed in the pathway. Now in the next step, again a water molecule is added, and the enzyme required for this step is 6-phosphogluconolactone hydratase. So I'll just write hydratase over here. Yes, so this is the enzyme which catalyzes the step for the conversion of 6-phosphogluconolactone to 6-phosphogluconolactone. gluconate. Okay, 6-phosphogluconate. So this is the next intermediate which is formed. In the next step, we have 6-phosphogluconate. I'll just write that again. We have 6-phosphogluconate which undergoes another dehydrogenation reaction, and the enzyme which is required is 6-phosphogluconate dehydrogenase. Again, for the step which involves removal of hydrogen, a molecule of NADP is required. NADP accepts the hydrogen from 6-phosphogluconate to form NADPH, again which is an important product of this phase, oxidative phase. And at the same time, there is removal of a carbon dioxide. So initially what you would have seen is you have a 6-carbon containing compound which is 6-phosphogluconate. With the removal of a carbon dioxide, one carbon is removed from the structure, leading to the formation of ribulose 5-phosphate. Now ribulose is a pentose sugar. It contains 5 carbon atoms, and the functional group present in this carbohydrate is a keto group. So it's a keto pentose sugar. So ribulose 5-phosphate is the last product which is formed at the end of oxidative phase. So with this, we are done with the oxidative phase. Remember, there are only irreversible steps, and the main purpose of it is generation of NADPH. And this particular phase is more prominent in the tissues which require NADPH for the production of various important compounds, starting from fatty acids, cholesterol, bile acids, as well as detoxification of various drugs. Now we'll move on to the next phase, which is non-oxidative phase. In here we have reversible steps and the formation of pentose sugars and hexose monophosphates required. So imagine now in the previous step I just spoke about how glucose or glucose 6-phosphate is converted to ribulose 5-phosphate. Now imagine this reaction has occurred 3 times. So you have 3 molecules of glucose which has undergone the oxidative phase to give you 3 molecules again of ribulose 5-phosphate. Same here and same here. Now you have 3 molecules of ribulose 5-phosphate entering into non-oxidative phase. So let's have a look at what really happens when these 3 molecules enter into non-oxidative phase. So I'll just write those 3 substrates over here. Ribulose 5-phosphate, and the last one here. So 2 of the ribulose 5-phosphate gets converted to an epimeric form by the help of the enzyme epimerase. So what do you mean by epimerase? Again, these are reversible reactions. So epimers are nothing but substances which differ with respect to a single carbon atom other than the penultimate carbon atom with respect to the arrangement of the hydroxy and the hydrogen atoms attached to that carbon atom. So generally the structure of ribulose 5-phosphate is something like this. Ribulose 5-phosphate is a keto sugar, so you have a keto functional group present, and then you have something that looks like this. This is the last carbon atom, which is CH2OH. So this is the structure of ribulose 5-phosphate. Now, in the step which is catalyzed by epimerase, what happens is you have an epimer form of ribulose 5-phosphate formed next, known as xylulose 5-phosphate. Again, xylulose 5-phosphate is a keto sugar, but xylulose is supposed to be a C3 epimer form of ribulose 5-phosphate, meaning at the level of the third carbon atom, there is a change in the orientation of the hydroxy group as well as the hydrogen group. So if this was the structure of ribulose 5-phosphate, the structure of xylulose 5-phosphate would be something like this. The keto group would remain. Only the third carbon atom, the orientation of hydroxy and hydrogen would change, where you would have hydrogen towards the right hand side and hydroxy towards the left hand side, and then the remaining structures are the same. So which is why xylulose 5-phosphate is said to be a C3, or xylulose is said to be a C3 epimer of ribulose. Now we have the formation of xylulose 5-phosphate, which is a ketopentose, and at the same time there's one more ribulose 5-phosphate over here. It gets converted to its isomeric form, functional isomeric form, with the help of the enzyme isomerase. So if ribulose is a ketopentose, the corresponding aldopentose is known as ribose. Now, what really happens is, especially in organs or tissues where the primary aim is, multiplication of cells. So whenever there is a lot of cells multiplying at a very rapid rate, especially in the bone marrow, intestinal mucosal cells and the tumor cells, those cells require a lot of nucleic acid for multiplication. So ribose 5-phosphate plays a major role in the formation of purines and pyrimidines, which are utilized in nucleic acid formation. Hence, in those tissues which require a lot of nucleic acid, the entire pathway stops at ribose 5-phosphate. And in the other tissues where the main aim is to generate only NADPH for the synthesis of fatty acid, cholesterol, etc., the pathway continues after that and then leads to the generation of glucose 6-phosphate, which can be reutilized in the oxidative phase. So now we'll just have a look at what happens next. We have xylulose 5-phosphate, which is a ketopentose, and we have a ribose 5-phosphate, which is an aldopentose. So both these two react together and the step is catalyzed by trans-ketolase, which requires a coenzyme known as thymine pyrophosphate, TPP. So thymine pyrophosphate is derived from the vitamin B1 or thymine. Hence, whenever there is a deficiency of vitamin B1 or thymine, this particular step may get inhibited or may not occur at all, leading to the accumulation of the pentose sugars in those cells. So next, what really happens is a keto group is transferred from the ketopentose to the aldopentose. The enzyme trans-ketolase, as the name suggests, is involved in the transfer of a ketose group. So during that transfer, what is seen is a 2-carbon containing entity from xylulose 5-phosphate is transferred to ribose 5-phosphate. So in the next step, what we get is, with the removal of 2 carbon atoms here, we get an aldotriose known as glyceraldehyde 3-phosphate. The keto group would have been shifted to ribose 5-phosphate. That's why we are left behind with an aldose sugar. And on the other hand, we get a 2-carbon atom which gets attached here to ribose 5-phosphate. So we get a ketoheptose known as pseudoheptulose 7-phosphate. So you can see the total carbon atom which has entered into the reaction will remain the same. You had total of 10 carbon atoms initially, and with the product you still have 10 carbon atoms, 3 plus 7. Now in the next step, these two react. Glyceraldehyde 3-phosphate and pseudoheptulose reacts together with the help of the enzyme trans-aldolase. So during this step, it is said that 3 carbon units from pseudoheptulose gets transferred to glyceraldehyde 3-phosphate. So what happens is the keto group also gets transferred here, leading to the formation of a ketohexose. You have 3 carbons from here getting added to glyceraldehyde along with the keto group. So you get a ketohexose, which is fructose 6-phosphate. And remaining, if you just remove 3 carbon atom from the 7 carbon containing compound, you're left with 4 carbon atoms. So you get erythrose 4-phosphate. This is a aldotetrose. Again, the total carbon atom remains the same. You have 6 plus 4. Okay. So in the next step, I'll just write all those intermediates again. Fructose 6-phosphate and erythrose 4-phosphate, and then we also have xylulose 5-phosphate, which was formed by the third ribulose 5-phosphate. So what really happens to fructose 6-phosphate is it does not participate further in the reaction. Rather, it gets converted to glucose 6-phosphate, which is a functional isomer form of fructose. Glucose is a functional isomer of fructose. And the enzyme which catalyzes the interconversion of both these functional isomers, fructose and glucose, is known as phosphohexose isomerase. So you can see here how you have a hexose monophosphate which is formed, and this may enter into glycolysis. Hence this is why HMP is known as a shunt. Okay, the hexose monophosphate is shunted into glycolysis, or it could be used to form NADPH by entering into the oxidative phase again. So this is what really happens to fructose 6-phosphate. On the other hand, we have erythrose 4-phosphate and xylulose 5-phosphate, which reacts again. And the enzyme required is transketolase. Thymine pyrophosphate is a coenzyme required for the activity of transketolase. And during this step, a 2-carbon entity is transferred from xylulose 5-phosphate to erythrose 4-phosphate. And at the same time, even a keto group is also transferred, leading to the formation of a 6-carbon keto group over here, that is fructose 6-phosphate. And if you minus 5 minus 2, you get a 3-carbon containing compound, that is an aldose sugar, that is glyceraldehyde 3-phosphate. Again, fructose 6-phosphate can form its functional isomer, that is glucose 6-phosphate. And glyceraldehyde 3-phosphate can enter into gluconeogenesis, where it can form glucose 6-phosphate. Gluconeogenesis is a pathway by which new glucose is formed from non-carbohydrate sources. So glyceraldehyde 3-phosphate can also form glucose 6-phosphate, one of the intermediates in the pathway of gluconeogenesis before the conversion to glucose. So this is how you have, hexose monophosphates in the form of glucose 6-phosphate formed at the end of non-oxidative phase. That's how the entire name of the pathway has come into being. Now coming to the significance, as I've already told you, there are two major products formed. One is a pentose sugar, which was ribose 5-phosphate, and the other was NADPH. Now ribose 5-phosphate is known to form an important product known as phosphoribosyl pyrophosphate, PRPP. Now this PRPP may be utilized for the synthesis of purines and pyrimidines. Purines and pyrimidines are nitrogenous bases, and those bases, nitrogenous bases, are required for the formation of DNA and RNA, which make up the genetic makeup of a human being. So this is the role of ribose 5-phosphate in the formation of nucleic acid. Now, there are certain coenzymes also that can be formed from ribose 5-phosphate because those coenzymes contain a purine such as adenine. So example of coenzymes which contain purines are ATP, NAD and FAD. All these contain adenine. Even coenzyme A also contains adenine. So ribose 5-phosphate has two major roles. The next one is production of NADPH. So NADPH is required in the reductive synthesis, especially of fatty acids, cholesterol and steroid hormones. This occurs mainly in the liver, and fatty acids can also be synthesized in the lactating mammary gland as well as the adipose tissue. The next one is NADPH plays an important role in the detoxification of many drugs, especially in the liver. There is an enzyme system known as cytochrome P450 enzyme system present in the microsomes of the liver. This particular enzyme system, which is involved in the detoxification of various drugs, is present in the liver and requires NADPH. The next one is, it keeps glutathione in the reduced form. Now, what is glutathione? Glutathione is a biologically active peptide. It's a protein, it's a peptide which contains 3 amino acids. One is glutamic acid, the second one is cysteine and glycine. It's also known as gamma glutamyl cysteine glycine. So glutathione has various roles in the body, and the first and the most important role of glutathione is to detoxify certain free radicals. Now, at various points in our body, there are free radicals formed due to certain reactions that occur, metabolic reactions that occur. So one of the most major free radical which is formed is hydrogen peroxide, and glutathione helps detoxify hydrogen peroxide. Now, in order for glutathione to detoxify hydrogen peroxide, which is a free radical, it is important that the glutathione is in reduced form. Now, very often, the reduced form of glutathione is written as GSH. So I'll just show you exactly what really happens. Imagine there is a lot of hydrogen peroxide formed in the body. This is a free radical. The main aim is to detoxify this and form water, which is very safe to the body. Now, how does this detoxification occur in the presence of glutathione? So you need reduced glutathione. I'll write it as GSH. 2 molecules of reduced glutathione is required. And because of the reduced glutathione, hydrogen peroxide gets converted to water and glutathione gets converted to its oxidized form. So initially it was in the reduced form, it gets converted to its oxidized form, which is often denoted as GSSG. And the enzyme which is required for this is peroxidase. Now, it's not enough that this entire reaction stops at this part. It is important to regenerate the glutathione so that it's in its reduced form again. So the reduction of oxidized glutathione or the regeneration of reduced glutathione occurs with the help of NADPH. And this NADPH is generated through HMP pathway. This is one of the sources of NADPH. However, you may have other sources of NADPH production, especially in case of extra mitochondrial isocitrate dehydrogenase, and even in the presence of malic enzyme which converts malate to pyruvate. Those are other sources where NADPH can be formed, but this is one of the major sources. And because NADPH gives a hydrogen to oxidized glutathione, it gets converted to NADP, and you get your reduced glutathione once again. The enzyme required for this is glutathione reductase. So this is how NADPH helps maintain glutathione, especially in the reduced state. Now, reduced glutathione is very important to certain cells in our body or certain tissues, such as the RBCs, where a lot of free radicals can be formed, the cornea and lens, because these tissues are also exposed to oxygen, and exposure to oxygen can result at some point, formation of hydrogen peroxide, which is the major free radical formed. So reduced glutathione helps maintain the structure, the stability and the integrity of RBCs, cornea and the lens. It is also said that the presence of reduced glutathione can also maintain the transparency of the lens. The next and the most important function of NADPH is in phagocytosis. Phagocytosis is a process by which neutrophils or macrophages, they consume bacteria and try to kill it, especially when a person is exposed to some kind of bacterial infection or viral infection, etc. So just imagine this is a neutrophil or a macrophage which helps in phagocytosis. So it is said that whenever the neutrophil or the macrophage engulfs the bacteria or any foreign substance or object or entity, imagine this is the bacteria which is present and it has been engulfed by the neutrophil. Now what really happens during that time is oxygen which is present within the cell is utilized. There's a lot of oxygen which is utilized, which is why this is called as a respiratory burst. So oxygen reacts with NADPH, which is formed during HMP pathway. Now this is another cell or another tissue where NADPH is synthesized. So NADPH reacts with oxygen and forms a free radical that is known as superoxide. And the enzyme system involved is NADPH oxidase. So you have a superoxide which is formed. This superoxide is converted to another free radical, which is hydrogen peroxide. And the enzyme which is required for the conversion of superoxide to hydrogen peroxide is superoxide dismutase. This is a very important antioxidant. Antioxidants are substances which fight against free radicals. Then you have hydrogen peroxide which is formed. Hydrogen peroxide is further converted to another free radical, that is hypochlorous acid. And the enzyme required for this is myeloperoxidase. Now you can see there are three free radicals formed. You have a superoxide, you have a hydrogen peroxide, and you have a hypochlorous acid. Now you see there's so much of free radicals, and you might wonder what really does these free radicals do? Does it cause damage? No, it doesn't cause damage at all, but it causes damage not to the cell but to the bacteria which is present within the cell. So all these three free radicals, hydrogen peroxide, superoxide and hypochlorous acid, acts against the bacteria and kills it. So this is one of the few instances where free radicals has a beneficial effect, where it is utilized for killing bacteria that a person or an individual is exposed to. So this is about phagocytosis and the role of NADPH during the process. The next one is, I've already mentioned, RBCs are basically maintained or they're stabilized with the help of reduced glutathione. That is one important thing, how the RBC membrane is maintained. Other than that, you know, RBCs contain hemoglobin, and hemoglobin contains iron, which is usually in the reduced form. Reduced form is Fe2+. So at certain points, what happens is, maybe in the presence of drugs or maybe some kind of, phenomena, the iron can get oxidized to Fe3+. The presence of Fe3+ is quite bad and can lead to the formation of methemoglobin, where the oxygen carrying capacity of hemoglobin decreases. So this is really bad. In order to prevent this, we have NADPH, which can convert the oxidized form back to the reduced form, which is beneficial to any individual. So remember, NADPH's role in RBCs is mainly to keep glutathione in the reduced state and also to make sure that the iron which is present in hemoglobin is also in the reduced state. Moving on to the regulation. Now the regulation can be studied under two depending on the phases of HMP pathway. So under oxidative phase, you have one set of regulation, and under non-oxidative, you have another. So there are two main enzymes. Glucose 6-phosphate dehydrogenase is the main regulatory enzyme or the key enzyme of the pathway. And it is said that the NADPH and NADP ratio is very crucial in determining the activity of these two enzymes, glucose 6-phosphate dehydrogenase and 6-phosphogluconate dehydrogenase. So it is said that whenever there is a increase in NADPH, NADP ratio, the enzyme glucose 6-phosphate dehydrogenase gets inactivated. So it gets inhibited. So HMP won't occur. Whereas whenever there is a decrease in the ratio NADPH, NADP, HMP pathway will occur. So this is what I just mentioned in the table. NADPH will inhibit, because the ratio increases, will inhibit glucose 6-phosphate. Hence, there will be inhibition of HMP pathway. The pathway won't occur. Whereas a decrease of NADPH-NADP ratio, which is governed by an increase in NADP+, will activate glucose 6-phosphate and will allow HMP pathway to occur. So when does this ratio fluctuate is the next question. So generally, it is said that there's an increase in NADPH-NADP ratio, usually in diabetes mellitus and starvation. These are the two conditions where you can find an increase in NADPH-NADP ratio. Whereas a decrease in NADPH and NADP ratio can occur during the well-fed state. So NADPH-NADP ratio is only concerned with the rate-limiting enzyme, which is glucose 6-phosphate dehydrogenase. Whereas hormones can also influence the activity of these two enzymes. We have insulin and glucagon. Insulin is usually released during the well-fed state. So during the well-fed state, insulin is secreted by the pancreas. It's a hypoglycemic hormone, and it is known to activate these two enzymes, leading to the occurrence of HMP pathway. Whereas glucagon, it's a hyperglycemic hormone. Glucagon is released especially during starvation. So again, and even in diabetes mellitus, again, glucagon can inhibit those two enzymes, leading to the inactivation of the HMP pathway in starvation. So this was it about the oxidative phase regulation. Whereas the next one, that is non-oxidative phase, depends entirely on the availability of substrates. So if you have pentose sugars available, you can have the reactions occurring in the forward direction where you have hexose monophosphates formed in the end. So the availability of substrates in the form of pentose sugars is very important for the non-oxidative phase to occur. Now, coming to the last part, which is the disorders linked with HMP pathway, we have two main disorders. The first one is quite crucial and important. It is known as glucose 6-phosphate dehydrogenase deficiency, commonly seen in Africa and even in the Middle East to some extent. It is an X-linked recessive disease, meaning that males are mostly the ones who are affected, whereas females end up being carriers. And the clinical manifestations, which include hemolytic jaundice and hemolytic anemia, where the RBCs tend to, get lysed, leading to breakdown of the RBCs. So you have anemia that happens, there's decrease in the RBC count, and because of increased hemolysis, there's also hemolytic jaundice or prehepatic jaundice. So those manifestations occur especially when an individual is exposed to oxidative stress. Now, what do you mean by oxidative stress? Generally, in a normal person, if you have a look at the amount of free radicals that are formed and the amount of antioxidants that are present to fight against free radicals, you will see that both these entities are almost equal. There's a fine balance in the production of free radicals and in the presence of, in the amount of antioxidants which are present. So there's a fine balance between the two in a normal individual. In a condition like oxidative stress that can happen during infections or during exposure to drugs, what really happens is you will have a lot of free radicals which is formed that cannot be fought or detoxified by the available antioxidants in the body. So that is oxidative stress, where the amount of free radicals is quite high, which cannot be destroyed or detoxified by antioxidants. So that is oxidative stress. So generally, individuals with G6PD deficiency, they are asymptomatic. The clinical, appearance of hemolytic jaundice and anemia comes only when the individual is exposed to oxidative stress in an event of infection or exposure to drugs. So the main aim here is there's a decrease in NADPH production, and because of that, hydrogen peroxide, which is a major free radical, cannot be detoxified. Clinical features I've told you. So these are the precipitating factors. These are the factors which can cause oxidative stress. So we have infections. Any infection can cause oxidative stress. Exposure to certain drugs. I generally use this particular help or tool to remember the drugs that can cause G6PD deficiency, hemolytic anemia. So the triple A's include anti-malarials, drugs used in the treatment of malaria, which is primaquine or chloroquine, etc. Then we have antibiotics, certain antibiotics, such as sulfamethoxazole, and analgesics or antipyretics also. These are used in the treatment of fever or high temperature, like acetonilide. So these are examples of drugs where a person who has G6PD deficiency can start exhibiting symptoms. And last is fava beans, also known as broad beans. Intake of these beans can cause the clinical features to appear, and the condition is known as favasm. In addition to all these clinical features, it's also seen that the individuals who have G6PD deficiency also develop resistance to falciparum malaria. That is a parasite that is involved in causing malaria. So what happens is it is generally seen that because of hemolytic anemia, where RBCs are destroyed in G6PD deficiency, it is seen that the lifespan of the RBCs from 120 days, which is the normal lifespan, is reduced to quite a less duration. So what happens is with the reduction in the lifespan of the RBCs, the life cycle of Plasmodium falciparum cannot exist, because the parasite requires a certain amount of days to grow and proliferate. So that is not being able to happen because the lifespan of the RBCs are reduced. So I'll just show you what really happens in an RBC, especially in a G6PD deficiency. So imagine this is an RBC and you have oxygen present in the RBCs. Now, in an individual with G6PD deficiency, this reaction won't occur, where glucose 6-phosphate is converted by glucose 6-phosphate dehydrogenase to 6-phosphogluconolactone. This reaction won't occur. So as a result of which, NADPH, which is produced, doesn't happen. There's no NADPH which is produced. And NADPH, you know, is utilized for detoxification of hydrogen peroxide, which is a major free radical. Now, how did that major free radical, how is it produced in the RBCs? So especially in G6PD deficiency, what happens is in an event of oxidative stress, especially when an individual is exposed to infection or drugs, as I've just told you, oxygen can get converted to hydrogen peroxide. And generally, NADPH is required for the detoxification of hydrogen peroxide to water. How? That is simply by this reaction. NADPH is required to maintain glutathione in the reduced state. So I've already mentioned these reactions earlier. And reduced glutathione is required for the conversion of hydrogen peroxide to water. So because of the non-availability of NADPH, this whole part where reduced glutathione is not formed, because of the non-availability again of reduced glutathione, hydrogen peroxide is not detoxified in a G6PD deficient individual. Hence, the levels of hydrogen peroxide will increase and this can damage the RBC membrane, causing a break in the membrane, and the RBCs will get lysed and hence that can lead to anemia. So this was it about how G6PD deficiency can cause a lot of harm, especially in an event of oxidative stress. Now, the main biochemical diagnosis would be mainly by the measurement of the deficient enzyme in RBCs. So if you measure the activity, you might find it is reduced or completely absent. And the management would be to avoid exposure to the precipitating factors. So infections, I know it is very difficult to avoid, but more or less these individuals can be vaccinated, especially from common infections like influenza, etc. and hepatitis. And then symptomatic treatment of the hemolytic anemia is also important, especially with the blood transfusion that can be given to the patient. The next disorder is thymine deficiency. I've told you earlier when I was discussing the reactions of non-oxidative phase, I told you that transketolase requires a coenzyme which is thymine pyrophosphate, and it is derived from vitamin B1 or thymine. So in case of thymine deficiency, again, the whole activity of transketolase will be reduced, as a result of which there will be accumulation of pentose sugars, especially in the cells where the HMP shunt or the HMP pathway occurs. Very often, thymine deficiency is referred to as beriberi. And yes, there will be accumulation of pentose sugars in the RBCs. And in order to find out whether really this is due to G6PD deficiency or due to thymine deficiency, the best way to distinguish what is the cause for accumulation of pentose sugars, that would be just by measuring the transketolase activity in the RBCs. So whenever there is thymine deficiency, obviously there will be a decrease in the enzyme activity. Treatment would be just to give thymine to the patient, administer thymine to the patient. So with that, we are done with the topic of this session. So just to briefly summarize, definition HMP pathway is mainly involved in the synthesis of NADPH as well as pentose sugar in the form of ribose 5-phosphate. The main sites are mainly the liver, lactating mammary gland, the adipose tissue, testes, etc., which are involved in the synthesis of fatty acid, cholesterol, and various other compounds. Subcellular site is mainly the cytoplasm, and the key enzyme is glucose 6-phosphate dehydrogenase. Starting material, of course, could be either glucose or glucose 6-phosphate. Then the important products of the pathway, ribose sugars, are mainly utilized for the formation of purine and pyrimidine bases, which in turn can be utilized for the synthesis of DNA or RNA or also certain coenzymes. And NADPH is utilized for several biosynthetic reactions such as synthesis of lipids, fatty acids, cholesterol, bile acids, then detoxification of drugs, and several other purposes. So two phases are there, oxidative phase and non-oxidative phase. Oxidative phase is an irreversible set of reactions that occur there, and the main aim of the oxidative phase is the generation of NADPH. Whereas the non-oxidative phase mainly comprises of reversible steps, and the main aim here is the formation of ribose sugar, that is a pentose sugar, or the hexose monophosphate. Yes, there's no energy generated unlike the other oxidative pathways of glucose such as glycolysis as well as citric acid cycle. And regulation is at the level of two phases, that is at the oxidative phase, two main enzymes are regulated, that is glucose 6-phosphate dehydrogenase and 6-phosphogluconate dehydrogenase. These two enzymes are regulated by the ratio, that is NADPH NADP ratio, as well as hormones such as insulin and glucagon. Whereas the non-oxidative phase mainly relies on the availability of substrates. And lastly, I spoke about the disorders. There are two disorders. We have G6PD deficiency and Thymine deficiency. So that's all for this session.